My name is Paul Limburg, and I serve as Chief Medical Officer for Screening at Exact Sciences. It's a pleasure to join this QIW panel to discuss innovative ways to leverage new technology to advance cancer screening. From an overall public health perspective, we all recognize that effective screening can reduce both cancer incidence and mortality, but remains underutilized in the United States. At present, national guidelines only recommend screening for a minority of target organ malignancies, with approximately 70% of all incident cancers having no standard of care screening tests. Unfortunately, detection of these cancers is often delayed until after symptom onset, when the disease process is relatively advanced and treatment options are both more limited and less effective. Late-stage diagnoses of unscreened cancers are more common in vulnerable populations, emphasizing the need for better screening solutions that enable equitable access for all populations. To frame the lung cancer challenge more specifically, we know this disease is the leading cause of cancer death for both men and women in this country, in part because most patients present with advanced stage disease at the time of diagnosis. And while approximately 15 million people meet criteria for low-dose CT screening, fewer than 6% of screen-eligible adults are adherent with existing guideline recommendations. Increasing participation in lung cancer screening will no doubt save lives, since early detection is associated with improved survival, as shown here by estimated 5-year survival rates of 60% on the far right for local, 33% in the middle for regional, and only 6% for distant metastatic disease. While advances in therapy will also contribute to reduced lung cancer mortality, our biggest opportunity to disrupt the status quo is through downstaging disease at the time of initial presentation, using novel technologies and population-level strategies to enhance both screening assay performance and patient engagement in lung cancer early detection. One way to expand the reach of lung cancer screening is to change the screening paradigm from single organ to multi-organ early detection. By considering cancer prevalence in aggregate, the number needed to screen, shown on the y-axis, shifts from impractical and cost prohibitive to clinically actionable and financially feasible. Applying this tumor-agnostic approach would also afford the potential to screen detect less common malignancies, such as esophagus, pancreas cancer, others, that currently fall outside of existing guideline recommendations. Encouragingly, the biology of carcinogenesis appears to be well aligned with the concept of multi-cancer early detection, since many of the key cellular pathways and promising candidate biomarkers are shared across organ sites. Recent progress in genetic sequencing, methylation profiling, proteomic analyses, fragmentation, pattern recognition, and other laboratory methodologies provides the potential to identify common signals from anatomically diverse cancers using a liquid biopsy approach. Further investigation is ongoing to determine which class, or more likely classes, of cancer-related targets will yield the highest performance characteristics in blood-based assays, with the general consensus that a scalable multi-cancer early detection assay will need to be optimized for high specificity to reduce the number of false positive test results that may lead to additional diagnostic evaluation, excess resource utilization, and heightened patient anxiety. Shown on this slide is the draft workflow for CancerSeek, a multi-cancer early detection assay that's under development at Exact Sciences. Discussion of multi-cancer early detection screening eligibility is envisioned during any non-urgent primary care visit, with subsequent blood sample collection and biomarker analyses. For the projected 99% of patients who test negative, repeat screening would be recommended at an interval to be determined. Conversely, for the 1% of patients who test positive, follow-up evaluation with PET-CT imaging would be recommended to identify and localize any of the targeted cancers. This model was recently assessed in a large prospective study conducted within the Geisinger Health System patient population. The goals of the study were to detect cancers not found by standard of care in the real world, to pilot blood test and workflow in a large clinical population, and to manage patient care by delivering test results. With respect to the study design of DETECT-A, the blood test used was a prototype of CancerSeek. There was a quality standards board made up of multidisciplinary experts who reviewed the test results. And then for those who were deemed to need a positive follow-up, there was imaging that was recommended and performed, and then appropriate follow-up after imaging based on those results. With respect to the enrollment, over 10,000 women were enrolled onto DETECT-A between the ages of 65 and 75 years over about a 2-year time period. And the only exclusion criterion was that they could not have had a prior history of cancer. Over a 1-year follow-up period, 96 women in the study were diagnosed with incident cancer, including 24 cases diagnosed by standard of care screening, 26 cases detected first by the CancerSeek assay, and 46 cases diagnosed as a result of symptom-based evaluation. Summarizing these data, the DETECT-A study found that application of a multi-cancer early detection test resulted in a 2-fold increase in screen-detected cancers, while further demonstrating the feasibility and safety of this novel screening approach. With respect to lung cancer diagnoses, CancerSeek led to detection of 9 women with lung cancer, which was 3 times the number of lung cancer cases detected by standard of care screening methods. Moreover, 7 of these women did not meet established high-risk screening criteria for lung cancer screening and would likely have remained undiagnosed until after symptoms appeared. The other 2 women whose lung cancers were detected by a multi-cancer screening approach did qualify as high risk, but were not adherent with current lung cancer screening recommendations. Building on the promising proof of concept and early feasibility data for CancerSeek, the next steps in assay development for this particular approach will include initiation of an FDA registration trial and launch of a laboratory-developed test, which are planned in 2022, to expedite clinical access while simultaneously generating additional evidence based on real-world experience. With the projected availability of the CancerSeek assay and other multi-cancer early detection tests on the near-term horizon, this novel strategy holds promise for shifting clinical practice to a much higher proportion of screen-detected cancers and reducing the overall and lung cancer-specific public health burden by expanding the screening target population with the promise for equitable access, increasing participation rates, identifying other currently unscreened target organ cancers that can be diagnosed at earlier stages, thereby reducing treatment costs and improving clinical outcomes. In order to achieve this vision, collaboration will be essential to both realize and accelerate that full potential of multi-cancer early detection. And it has to include policy, patient advocacy, academic, other key stakeholder organizations across the cancer spectrum. Appreciate the opportunity to engage with this group today and to continue the conversation. Thank you.